Hi, my name's Brad Hoge. I'm one of the four professors who are going to be helping you this week, and I really apologize I'm not here today. I will be here in the morning, and my student Juan Gerardo Parra will be here today with you to help you figure out what it is I'm doing and what my procedures are. But I wanted to talk to you just for a few minutes to introduce myself and to tell you a little bit about the project that I'm working on. My research project is kind of hard to explain and kind of easy in some different ways. I use diatoms. Diatoms are single-celled algae that live in any body of water, so they're present in all of the bayous and lakes and ponds and swamps in the area. I use them as bioindicators. I don't study the diatoms themselves per se. I use them to tell me what's happening in these aquatic environments. How are they changing over time? Diatoms respond really well to changes in the environment, and their assemblages, in other words, the groups of diatoms that you can find in a given location, are pretty specific to that location, and any changes then that occur in a location can be sort of seen as the diatom assemblage changes. I started this project when the Harris County Flood Control District came to me over 10 years ago now and asked if there was a way I could do a research project at the Greens Bayou Wetland Mitigation Bank. And for the first 10 years of the project, I was looking at the assemblage at the Greens Bayou Wetland Mitigation Bank and comparing it to assemblages that I knew were stable and representative of a similar environment at the Anahuac National Wildlife Refuge. Now, this introduces a couple of things that have led to my current research. One was that I'm basically claiming that the assemblage at Anahuac is what the assemblage at the Greens Bayou Wetland Mitigation Bank will become as the wetland changes, as the soil and the bacterial community creates the conditions at the Greens Bayou that exist at Anahuac. Anahuac's not that close to Greens Bayou, and in fact, it's on the other side of Galveston Bay. So how do I have any confidence that they'll eventually reach the same climax community? Well, they're both within the Trinity River incised valley. As the glaciers retreated over the last 12,000 years or so, the Trinity River has retreated, and the Gulf of Mexico has flooded into the valley that the Trinity River had cut into the area when our shoreline was about 200 miles farther out to sea. We would have been about 200 miles inland 10,000 years ago. So the river carved a canyon that is then filled up over stages with sediments. You can still see throughout this watershed, the Trinity River, that the flooding that we experience is contained within this geologic watershed. So what I am doing now is looking for proof of that. I'm looking at what are called edge effects on the succession trajectory of diatom communities at Greens Bayou Wetland Mitigation Bank, meaning I'm trying to define what the other watershed assemblages are in the various bayous and rivers and streams within this incised valley, and how those diatoms can lead to the changes in the communities that we see at Greens Bayou over a period of time, over thousands of years. This requires major flooding events, and we've seen quite a few. Even in the last 10 years, I've seen Hurricane Ike, and there's clear evidence that it brought diatoms and other marine species actually into Greens Bayou Wetland Mitigation Bank. We've recently had flooding this year in particular, so this summer's evidence is going to be very valuable. And this is the case that I'm making, is that over time these extreme flooding events mix all of the assemblages within the Trinity River incised valley so that the climax community that a given environment will reach will be similar within the valley, even if they are separated from one side to the other or from the middle to one side, as we have with Greens Bayou and Anahuac. And I think we're starting to see this, but that's what you're helping me with this summer. You're each contributing samples that we can analyze from different watersheds throughout the area. I've collected samples and had students collect samples from numerous places, but the more I can find and the more I can help to identify, the more I can use a clustering analysis, a statistical analysis to truly define the various types of environments within the larger watershed. Right now it looks like we have 4 or 5 unique assemblages that can be attributed to small bayous and streams, larger rivers, swamps, and marshes and marsh ponds, such as Greens Bayou and Anahuac. So as I look at all of these surrounding watersheds and use statistical analyses to try to determine if I can actually define an assemblage for these different environments, I can then map how those diatoms get from those environments into the Greens Bayou Wetland Mitigation Bank. So you won't have enough time to really count enough diatoms, but hopefully you'll leave your samples with me and we'll continue to communicate. And eventually we'll have enough data to actually say how it is exactly that the assemblage at the Greens Bayou Wetland Mitigation Bank has managed to change from the time I started assessing it over 10 years ago to today, and how today it actually looks more like Anahuac than it did not only 10 years ago, but then it does any of the other environments within the Trinity River and Sized Valley. So I think we have a good story, but we need the evidence. It's all based on the data, and that's what you're helping me with. So I will see you in the morning, and I appreciate your help. And if you have any questions, hang on to them for me, and I will answer them to the best of my ability when I'm here tomorrow and the rest of the week. Thank you very much. I look forward to meeting you.